Okay, today's video discussion is meiosis, which is the nuclear division that reduces the number of chromosomes in the new cell. So if you know mitosis, you take a cell and make two cells with equal number of chromosomes. In meiosis, you have to reduce the number of chromosomes, and this is all due to sexual reproduction. So today is different than mitosis because we're discussing meiosis, and we're going to look at the stages of meiosis, the results, and actually look at how mitosis and meiosis are similar and different and the uses for both of them. So again, long process. If you stick with me by the end, you'll be rewarded by amazingness. All right, so meiosis, as said before, reduces the number of chromosomes. It is used for sexual reproduction because if you just went through mitosis and then joined those two cells, you would end up with double the number of chromosomes as the original cells and the original person, and therefore every generation we had would have double the amount of DNA, which would be problematic because by this point in our history of humans, we would have way too much DNA. So cells undergo meiosis to produce your gametes or your sex cells. So cells undergo interphase, same as mitosis. The cell cycle is the same except for that M phase, and there are basically two stages of division, meiosis 1 and meiosis 2, with no interphase in between, no DNA replication. So you start off the same, and it has 4 phases in each. So meiosis 1 is prophase 1, metaphase 1, anaphase 1, telophase 1, and cytokinesis 1. So each one of these corresponds to the phases of mitosis. There is a major difference that occurs in prophase 1, and that difference is the pairing of homologous chromosomes known as synapsis. So we're going to, again, like in mitosis, go through each phase and determine what happens and the results of each phase. So prophase 1, similar to prophase in mitosis, the spindle fibers form and the nuclear envelope disappears. Now a major difference is the homologous chromosomes pair up. Homologous chromosomes are chromosomes that have similar genes on them. You get one from your mom and one from your dad. So a pairing of homologous chromosomes are known as a tetrad because you basically have 4 chromatids, tetra 4, and it's a pairing of homologous chromosomes. Now during this pairing, crossing over can occur. Crossing over is shown in the picture, and it's when the two chromosomes get so close that they actually swap DNA. And this increases genetic variation, which actually is a good thing, because if we didn't have genetic variation, if we didn't go through meiosis and have differences like this, then would all look the same. And we definitely do not want to all look the same, because that would be boring and would all have the same personalities and everything, and that'd just be bad. So that is prophase 1. Metaphase 1, the homologous pairs line up at the center. So you have 2 lines, and they line up at random. And the spindle fibers, again, attached to each homologous chromosome, crossing over can occur, and they do line up at random, which is known as independent assortment. Basically meaning that you don't have all your mom's chromosomes on one side and all your dad's chromosomes on the other side. They mix and match. It doesn't matter what one pair does, what the other pair does. It's kind of like if I paired you up boy-girl and then had you line up, it wouldn't matter if if a boy went on one side and a girl went on the other side, to the next pair. They line up independent of one another. It's the law of independent assortment, that each homologous chromosome, doesn't matter what the other homologous chromosomes are doing, they'll just line up. So in anaphase 1, the homologous pairs are separated. Very similar to anaphase and mitosis, except that you're not breaking up chromatids, you're breaking up homologous chromosomes. And then telophase 1, chromosomes reach the opposite ends, the nuclear envelope starts to reform, cleavage furrow or the cell plate pinches off and completes cytokinesis after that, and you end up with 2 haploid cells that are not identical. And haploid, remember, means no homologous chromosomes. because you just split up the homologous chromosomes into two separate cells. And you have half the number of chromosomes. So already each cell in a human has 23 chromosomes. But each chromosome is still in that double-stranded form. Now, if we move on to meiosis 2, we have to make sure we don't go through interphase and DNA doesn't duplicate, because each chromosome is already 2 strands. So no DNA duplication. Meiosis 2, as said, DNA is not copied and occurs in each cell. So therefore, both cells that were formed in meiosis 1 go through meiosis 2. And the 4 phases should look familiar by now: the PMAT, prophase 2, metaphase 2, anaphase 2, telophase 2, and cytokinesis 2. In some species, the nuclear membrane reforms after meiosis 1, but there is no real interphase regardless, and honestly, in animals, in humans, we do not reform because that's kind of a waste of energy. So prophase 2, a new set of spindle fibers form, because remember, those had to go away in order for the cleavage fiber to come in, and the chromosomes start to move towards the middle. Then in metaphase 1, all chromosomes line up in the middle, exactly like in mitosis, so that there's one line. And then in anaphase, they separate the chromatids. So back to the chromosome is just densely packed DNA, because each one of these had 2 chromatids. Now you're separating the chromatids. And then in anaphase 2, the nuclear envelope reforms and the cleavage furrow, followed by cytokinesis. So then you end up with 4 haploid daughter cells that only have 1 strand of DNA. Very, very important to realize. 1 strand of DNA versus where at the end of meiosis 1, each chromosome was double-stranded. So the results, you have 4 new different cells. They each have half the number of chromosomes as the original cell. And in animals, these produce your gametes, and occurring only in the reproductive organs. And in male gametes, 4 sperm cells are produced, and it's known as spermatogenesis, the production of sperm cells. In females, interestingly, you only make 1 egg and 3 polar bodies, because during the cytokinesis, the cytoplasm is divided unequally. So you get 1 large egg and then 3 small eggs. That's 3 small polar bodies. This is known as oogenesis, and the 3 polar bodies generally stay around to give the egg protection and nutrition once it gets fertilized. If it doesn't get fertilized, those polar bodies go away. So if you compare mitosis and meiosis, number of phases, 4 versus 8, results, mitosis, 2 identical cells with the same number of chromosomes. In meiosis, you get 4 different cells with half the number of chromosomes, and a difference in these steps. In mitosis, there's no pairing of homologous chromosomes. In meiosis, there is pairing and independent assortment. And then problems that could occur is for mitosis, if the chromatids don't split, so that there's no chromosome splitting. In meiosis, if the chromosomes don't split or the pair of chromosomes don't split, which that usually results in the chromosome disorders that we looked at before, like Down syndrome. Down syndrome, you have an extra chromosome. So if during meiosis, one of the pairs of chromosomes didn't separate, one of the cells would end up with 2 of the same chromosome. So this follows the number of chromosomes and the amount of DNA through each phase, which clarifies the difference between the double strand versus chromosome number. So if you look at these numbers closely, for mitosis, your final results, you end up with 2 cells with 46 chromosomes and, say, 5 grams of DNA. Obviously, you don't have 5 grams of DNA, but we're just using that number for the sake. And then meiosis, you have 4 cells with 23 chromosomes and 2.5 grams of DNA. So you actually also have the amount of DNA per cell, because you have half the number of chromosomes by the end of meiosis. But if you look at after telophase 1, in meiosis, you have 46 chromosomes to 23 by the time cytokinesis ends, but you in each cell have 5 grams of DNA because you have those double strands. So that's to show you the difference between the chromosome and amount of DNA through the phases. So that is the complete cycle and cell division and all of cell reproduction. So if you have any questions, write them down now and come in with questions. We're hopefully going to be able to get all of these steps in order and organized, but focus mostly on the complete picture. Don't get into the nitty-gritty numbers that were at the end. Those are just to help you recognize the differences. Focus on the phases and the need for all of these. Major difference mitosis is for most of your cells to reproduce to replace cells that you've lost, and meiosis is to produce egg and sperm cells for final reproduction of the organism.